Hello children, today we are going to learn very important questions of molecular basis of inheritance of genetics lesson class 12. These are important for CBSE board examinations as well as competitive exams. Okay, what is reverse transcription and explain how single stranded RNA of viruses give rise to double stranded RNA. RNA is of single strand and DNA is made of double strand. Okay, let's see that how RNA is like a single stranded RNA gives rise to double stranded RNA. Okay, reverse transcription is the process. In this process, synthesis of DNA from RNA using an enzyme called reverse transcriptase. In viruses, these type of enzymes are present in retroviruses. Okay, RNA is used as template for the synthesis of single stranded DNA. Then this is known as complementary DNA, also called as cDNA. Using the enzyme, this is done using the enzyme reverse transcriptase. Next question, what is inborn error metabolism? It is a genetic disorder which results in the production of defected enzyme required for catalyzing the conversion of one substance into other type of substance. This was discovered in Archibald Garrod in 1909. He found that in the black urine disorder, that is alkaptonuria, and this is also inherited disease. It is an inherited disease. And what is this alkaptonuria? And it is a genetic disease where the metabolic pathway for degradation of tyrosine and phenylalanine is disrupted. That means homogeneous acid, that is HGA, an intermediate of the pathway, builds up in the bloodstream of the patients and excreted via urine. That is, alkaptonuria is a rare condition in which person's urine turns dark brownish color or black color when it is exposed to air. Next question, what is one gene, one enzyme hypothesis? Okay, it is a hypothesis put forward by Beadle and Tatum in 1948, which proposes that a gene operates through its control on the synthesis of enzyme. The two workers obtained evidence from their work, and induced in auxotrophs and of Neurospora crassa, that is Beadle and Tatum in 1944. And what is retrovirus? Already we discussed. And any of the family of RNA which consists of reverse transcriptase enzyme and capable of making a complementary DNA of the viral RNA. Okay, and which then is integrated into the host cell, and host cell accept as its own and functions. And the family includes a number of significant pathogens, typically causing tumors and affecting the immune system, and example HIV. Children, expansion of HIV, human immunodeficiency virus, it's a pathogen and which causes disease AIDS, acquired immunodeficiency syndrome. It's a syndrome, means group of diseases. And besides human immunodeficiency virus, the virus that causes AIDS, and there are two other retroviruses that can cause human illness. One is called human T lymphotrophic virus type 1, that is HTLV1, and the other one is called T lymphotrophic virus type 2, that is HTLV2. Next question, name the type of operons on the basis of their induction and repression. Inducible operons example is lacoperon, and repressible example is tryptophan operon. Okay, and in which metabolic pathways do we find these inducible and repressible operons? Inducible operons are seen in the catabolic pathway. Catabolic pathway is the pathway where that complex component is broken down into simpler ones. And repressible anabolic is a anabolic pathway, that is the simple components from the complex one. Next question. What do you mean by proofreading DNA synthesis? What do you mean by proofreading activity of DNA polymerase? This is done by DNA polymerase enzyme. So DNA polymerase, these are the enzymes that build DNA in cell. Okay, during DNA replication means copying the new, and most DNA polymerases can check their work, proofreading, and with each base that they add. Whenever they add, they will check the sequence, and this process is called as proofreading. So DNA polymerase proofreading is a spell checking activity. It will check the base pair spellings. At simple, we have seen that sickle cell anemia, simple base pair change may give some mutations. So this DNA polymerase enzyme is a very important enzyme which will check the spellings, enables DNA polymerases to remove newly made nucleotide incorporation errors from the primer terminus before further primer extension. Before next one starts, this will check complete base pairs whether they are in the proper condition or correctly copied or not. And so this is the proofreading is done by DNA polymerase, and also prevents translation synthesis also. Translation, if it is required, it will proceed. If it is not required, that means if any error is there, it will stop translation. I think you know what is translation. Okay, conversion of RNA into protein, central dogma, you might have heard. We'll go to that part also next. What is contact inhibition? What is its significance in cell division? Contact inhibition is a process of arresting cell growth when cells are in contact with each other. When exactly arrest is required, when the cells are under enormous growth. Where do you see enormous uncontrolled growth? In the cancerous cells. Okay, as a result, once it is stopped, contact inhibit, then normal cells stop proliferating when they form a monolayer of the culture medium, culture dish. Okay, contact inhibition is a powerful anti-cancer mechanism that is lost in cancer cells. It's a very important process, children, to control the enormous growth of the cells in the cancer. How is DNA content in our diet digested in the body? DNA, nucleic acids consist of RNA and DNA, and in the food, of course, we eat cellular food and all. It is digested in the small intestine with the help of both pancreatic and small intestinal enzymes. And what pancreatic enzymes are called neuribonucleases, that means which will digest RNA, and deoxyribonucleases that will digest DNA, and they break down RNA and DNA respectively into a smaller nucleic acids for the absorption. Next question. What are genomic and complementary libraries? How can they be prepared, and what are its advantages? A genomic library is a collection of total genomic DNA from a single organism. The DNA is stored in a population of identical vectors, each containing a different insert of DNA. So the complementary DNA library represents the collection of only the genes that are encoded into the proteins by an organism. Children, you know that in the gene banks and all, they are storing samples, blood samples and tissue samples and all for stem cell therapy. So this can be used here. Next question. DNA polymerase and RNA polymerases differ in their requirements while functioning. How? That means DNA polymerase already we have seen, which will help in the polymerization as well as proofreading activity, while RNA polymerase is only polymerization activity. DNA polymerization as well as proofreading, but RNA polymerase only for the polymerization activity. And DNA polymerase insert nucleotides and repairs the mismatched pairing in the proofreading. Proofreading itself is like that. Like if any errors are there, if any mismatch is there, any obstruction is there, anything goes wrong, then DNA can identify that and check that and repair it also if possible. And DNA polymerase proofreading is a spell checking activity that enables DNA polymerases to remove newly made nucleotide incorporation errors from the primer terminus before the further primer extension and also prevents the translation synthesis also here. Children, sometimes even to kill the pathogens and all, we take antibiotics, no? And they also help in this processes, transcription and translation, not to which will block these activities in the viruses and bacteria, so that it will be cured, which will not allow bacteria or virus to spread in the body. It will die there, then and there itself, so the disease is cured. What are the main kinds of reproductions in viruses? Give examples. And lytic cycle T4 bacteriophase is one, and lysogenic cycle lambda phase is another one. These are the two kinds of reproduction in viruses. Next question. Differentiate between transformation and transduction in bacteria. Very important. Transformation is a transmission of DNA from dead bacterium to the living cell. Carrier organism is not required here because dead organism is introducing into the new bacterium. But transduction is in this case DNA is transmitted from one bacterium to another. Here both the bacteria are living, and through this is done through the bacteriophages, and in this virus is the carrier. You know that what is bacteriophage? When the virus attacks bacterium, bacteriophage is formed. This you learnt in class 11th. Now come to this one. What is central dogma? This is what I said. Conversion of DNA into RNA is by the process of transcription, and RNA into protein by the process of translation. Ultimately, DNA is converted into protein. Protein is the component which will express in the genes. Okay, and central dogma, the process of synthesis of proteins involves one of the central dogma of the molecular biology. This is very important. According to this, genetic information flows from nucleic acids to protein. Nucleic acids are DNA and RNA. Initially, DNA is converted into RNA by the process of transcription, and RNA is converted into proteins by process of translation. And we know that the DNA is stably inherited from the parents to offspring. So, according to central dogma, the flow of information takes place from DNA to RNA, RNA to protein. So, this is this you are having in your textbook also. You can go with that. Very, very important question. Next question. What is the category of virus that carries reverse transcriptase? I told you already, that is the retrovirus, which will help in the conversion of DNA conversion of DNA into DNA from RNA, and also this DNA get incorporated with the host cell for the further processes. Next question. What is the difference between the function of primase and DNA polymerase? Primase is an enzyme. It enzyme ASE at the end. If the term comes that is an enzyme. You know that it catalyzes the formation of primer, which is polynucleotide during DNA replication. It is starting block of this DNA replication. DNA polymerase is formed from formation of DNA during DNA replication, and proofreading is function. And some more facts I'll give in the next episode. Thank you so much, and all the best.